This is Suganti, Assistant Professor, Department of Biotechnology, Vistas, Chennai. In today's class, we are going to see about the basic DNA structure and function. We all know that DNA is a major macromolecule and it's carrying the genetic information. The small segment of DNA is to make the gene. The DNA is consists of two parallel strands it twisted together to form a double helix. We know that the DNA is a double helical model, and DNA molecule is wrapped around a protein, and it will form a chromatin thread. And these chromatin threads are present inside the chromosomes. And during this cell division, these chromatin threads are tightly coiled and into structure called chromosomes. So these chromosomes are present inside the cell nucleus. This is the double helical structure of DNA. So the sugar phosphate backbone is present on the outside. The base pairs adenine, guanine, cytosine, thymine. The four nitrogenous bases are present inside the double helix. And the direction is one in the two anti-parallel strand. One strand is 3' to 5' direction, and the opposite strand is 5', 3' direction. DNA contains the sugar phosphate backbone with the four variable bases. So this structure is called as nucleotide. So nucleotide is nothing but is made up of sugar, phosphate, and nitrogenous bases. So the components of nucleotides is nothing but base, sugar, and phosphate. This is the structural model of nucleotide. So in this you can see the phosphate group is attached to the sugar, deoxyribosugar is present in the DNA. So this phosphate and sugar are present in the outside helix, that is phosphate backbone of the DNA. And so this sugar, deoxyribosugar, is attached to the any one of the nitrogenous bases. It may be adenine, guanine, cytosine, and thymine. So we have seen that nucleotide is a combination of three things: phosphate, sugar, and base. And what is nucleoside means? Nucleoside is nothing but sugar and base. This is present in the outside phosphate backbone of the DNA. So this is the basic structure of purines and pyrimidines. We all know that cytosine and thymine are the example of pyrimidine. And uracil also grouped under pyrimidine. And the purines are adenine and guanines are the example of purine. So traditionally, the DNA sequence is drawn from the 5' to 3' end. The opposite strand is 3' to 5' end. So this is the primary structure of DNA. So in this, you can see the phosphates, sugar, and bases are attached together. Now we'll see the base pairing of the DNA. Okay, the base pairing of DNA is very, very important in the DNA structure. So inside the DNA, two strands of the DNA inside the bases are present. So inside the base, inside the strand, how the bases are connected. So it describes the Chargaff rule. Okay, so before that we should know what are the bases, nitrogenous bases. Purines are adenine and guanine are grouped under purines, and thymine and cytosine are grouped under pyrimidines. And we must know which base is paired to the which base. Okay, for example, guanine is always paired with the cytosine. Okay, with the help of 3 hydrogen bonds. Here you can see the 3 hydrogen bonds. Okay, but in the case of adenine and thymine, adenine is always paired with thymine with the help of the 2 hydrogen bonds. But actually, these hydrogen bonds are very weak bases. So this is the chemical structure of DNA. Here you can see this is sugar, and sugar is connected to phosphate, and phosphate is again connected to sugar. This is the outside helix. So this is one strand and this is one strand. You can see clearly. But here this one of the sugar is connected to guanine. So on the same way, on the opposite strand, the sugar is connected to cytosine. So likewise, adenine and guanine are joined by three hydrogen bonds, and adenine and thymine is connected by two hydrogen bonds. Okay, this describes the Chargaff rule. So Chargaff rule is nothing but adenine must pair with thymine. It won't mismatch and it won't go and mismatchly pair to cytosine. Likewise, the guanine is must pair with cytosine. The guanine it won't go and pair with thymine. Okay. And also the Chargaff rules also describe the amounts of given DNA molecule will be about the same. That means, for example, the percentage of adenine is 40% means automatically the percentage of thymine is also 40%. And then the guanine is 60% means the automatically the percentage of cytosine is 60%. So here they have given the percentage of adenine is 30%, the thymine is 30%, the guanine is 20%, cytosine is 20%. So here you you have to calculate adenine and guanine is 50%. So automatically another remaining 50% is thymine and cytosine. Now we'll see the function of DNA. Okay. We know that the DNA will code for the synthesis of the polypeptide, and many polypeptides will join together to form a protein. So which is responsible for the traits actually. So the DNA it will store the information needed for the development and functioning of all living organisms. The transmission of this information from mother to daughter cell is called gene transfer. Unlike other macromolecules, DNA does not serve a structural role in cells. It will play an important role in the um, transferring of the hereditary information. Now, apart from the function of DNA, the DNA is involved in the replication and mutation and recombination and other gene expression studies. So there are DNA is having the wide range of applications. So we must know the basic structure and functions of DNA to further studies like cloning and do the recombinant DNA technology and forensic like DNA fingerprinting and for genetic engineering. For all the things, the basic things about DNA is very important. And also this DNA fingerprinting is also called as DNA profiling, and for medical diagnosis and for agricultural application and human animal therapeutics. So whatever the thing, whether the plant or animal or human being, the DNA basic things about DNA is very important. Now we'll see there is a small quiz. So for example, if there is a 30% of adenine in DNA, what is the percentage of cytosine? So here they have asked in an opposite manner. So here you have to think like this, the same whatever I have shown in the earlier. So the adenine is 30% means cytosine is 20%. Next, for example, if I am giving the one strand of DNA like adenine, cytosine, adenine, thymine, cytosine, and guanine means you have to write the opposite bases. You can write right. Adenine means thymine and cytosine means guanine likewise. And also the ratio adenine and thymine is 1:1 and guanine is, that means the percentage of the adenine is equal to the percentage of thymine. Likewise, the percentage of guanine is equal to percentage of cytosine. This is like this is the checkpoint 2 to write the complementary basis. Again, they are asking about the percentage. This is very simple. This is the same. Again, they are asking about the percentage. Now here I have given the different terms which is used in the molecular biology, like from genes to chromosome. So here you have to rearrange these items in order to size. The largest first. So which one is largest one? Actually, the cell is the largest one. The next one is inside the cell, nucleus is present. So inside the nucleus, chromosomes are present, and inside the chromosome, what is present? Yeah, yes, DNA is present, and inside the DNA, gene is present, and inside the gene, the base pairs are present. So these are all the different things you have to know which one is small and which one is bigger one.